Good afternoon and welcome to the second meeting of the 2024 USDA Human Nutrition Research Center on Aging's Monday Seminar Series. Before introducing today's speaker, let me remind you to ask your questions for our speaker using the Q&A icon located on the webinar toolbar. Please do not use the chat box. You can add questions at any time during the presentation as they arise. There's no need to wait until the end of the presentation. And let me welcome today's speaker, Dr. Samaneh Farsijani. Dr. Farsijani is an assistant professor in the Department of Epidemiology at the University of Pittsburgh. She is a nutritional epidemiologist and a registered dietitian with extensive interdisciplinary research background ranging from basic biology to epidemiology. Dr. Farsijani's primary research focus is nutrition is the development of age-specific dietary recommendations to promote healthy aging using precision nutrition techniques, including omics data such as the gut microbiome and the metabolome, and non-omics data, including dietary intake. Dr. Farsijani also serves as the PI of the microbiome and inflammaging ancillary study of the SOMA cohort, SOMA being the acronym for the study of muscle mobility and aging. Dr. Farsijani is also exploring the role of chrononutrition, seeking to enhance dietary recommendations by incorporating not only the quantity and quality of food intake, but also the timing of consumption. And she will discuss this research today in her presentation titled Beyond Quantity and Quality, the Role of Chrononutrition in Health. And with that, let me turn the microphone over to Dr. Farsijani.
Thanks, Paul, for that introduction. And before I start, I'd like to say a special thanks to Dr. Sarah Booth for the invitation to speak at this seminar. It's a privilege for me to be here. So today, we're going to dive into the role of chrononutrition and how it ties in with muscle health, particularly in older adults. Well, as many of you might know, as we age, we lose muscle mass and muscle function that can lead to sarcopenia. And what you're looking at in this picture highlights how not just aging, but several factors, including nutritionally related factors such as unbalanced diet, inadequate energy intake, or inadequate consumption of dietary proteins, can play actually a big role in the development of sarcopenia. And this can lead us down a path to what's called unsuccessful aging, where we are more prone to frailty and other health issues. So as a result, several nutritional strategies have been developed or proposed to maintain muscle health in older adults. So in this slide, I have two examples of these dietary strategies. On the left, you see an example of a higher protein intake, around 1.2 grams per kilogram of the body weight, that has been linked to a slower decline in appendicular lean mass in community living older men and women, according to the findings from the Health ABC cohort. And turning to your right, there is another example of PUFA supplementation, which is derived from a meta-analysis of 10 randomized clinical trials. So basically showing that a daily supplementation with polyunsaturated fatty acids of more than 2 grams a day can significantly increase muscle mass in adults age 60 years and older. Well, there are other dietary strategies that have been proposed to maintain muscle health. As you can see, a couple of examples here, such as supplementation with essential amino acids, or vitamin D supplements. However, majority of these nutritional interventions are primarily focused on the quantity and quality of food or nutrient intake. And what's missing here is that how does the timing of intake impact muscle health that has remained unknown. And we have some recent evidence that they're showing that so the frequency and timing of food intake, which is referred to as chrononutrition, can impact body composition and overall health by entraining our body's circadian clock. So on your left, you see the architecture of our circadian system, which is made up of a central clock located in the brain. It regulates sleep and hormone production. It also receives signals. It actually influenced by light and dark cycles. And then we have these small clocks in other organs, in different parts of our body, for example, in the liver, in the gut, even in our muscles. And these small clocks are referred to as peripheral clocks. And they receive signals from the central clock and also these external cues. As you can see here, some of the examples, for example, physical activity level and food intake. And recent studies again have shown that circadian misalignment, which is the mismatch, this mismatch between the peripheral and central clock as a result of various factors. For example, exposing ourselves to light at night or irregular meal timing patterns, such as skipping breakfast or eating late at night, can lead to metabolic dysfunction, and it increases the risk of developing these cardiometabolic diseases, as you can see them here, such as obesity, diabetes, and hypertension. Now, the question is how meal timing can influence muscle health. Well, there is a wealth of research on meal timing for elite athletes in a sport nutrition aimed at enhancing their muscle mass function, their overall performance. As you can see, I've included some examples here, such as consuming meals earlier in the day to optimize nutrient partitioning, so that to increase muscle mass and function. Or the other one is consumption of proteins at regular intervals throughout the day to increase muscle protein synthesis. However, the significance of meal timing on muscle health in older adults is not well understood. Well, we have some small-scale human studies on healthy young adults that they've shown, for example, breakfast skipping is associated with lower muscle mass. However, these studies are not representative of the older adults population who, as I mentioned earlier, who are already experiencing the decline in their muscle mass and the decline in their muscle function. Well, we also know that chrononutrition encompasses a wide range of eating behaviors. As you can see them here, such as the time of first food intake, the time of last food or beverage consumption, eating duration, fasting duration, eating frequency, or late night eating. So a comprehensive assessment of all these chrononutrition behaviors is essential in order to understand how timing of intake contributes to muscle health beyond conventional nutritional factors. So what we did first, we characterized various chrononutrition behaviors and their 15-year trends among US adults using the nationally representative data from the National Health and Nutrition Examination Survey, or NHANES. Well, as you can see it here, in this project, we use data from 8 cycles of NHANES from 2003 to 2018, We included all the adults over the age of 19 who had two 24-hour food recalls. So our final sample for this project was 34,470 participants. On your right, you see the chrononutrition behaviors that we defined them based on the clock time of food intake, and we extracted them from the collected 24-hour food recalls, because again here, as you might be familiar with the 24-hour food recalls, we have the timing of intake. As you can see, we define the clock time of the first food or beverage consumption as the time at which the first food or beverage containing more than 0 calorie was consumed after midnight within 24 hours. And then we define the clock time of the last food or beverage intake as the consumption of any food or beverages containing more than 0 kcal before midnight, again within a day. And then we calculated the eating duration as the time elapsed between the first and last intake and expressed that as hours per day. Well, as for the eating frequency, we define that by counting the total number of self-reported eating episodes. But here we considered a new eating event whenever both the clock time and the self-identified meal changed. And finally, we identified late night eaters as those consuming any food or beverages containing more than 0 kcal between 9 p.m. and midnight, again within a day. Now, going over the results. So here you see various chrononutrition behaviors among young, middle-aged, and older adults. So what we observed was that among young people, they had a later first meal compared to the other age group, and they had a longer eating duration compared to older adults who had a shorter eating window, and they stopped eating earlier in the evening, as you can see here. We also looked at the patterns of energy intake according to the self-reported eating occasions, snack, breakfast, lunch, dinner. And as you can see, we observed this skewed pattern with about 36% of total calorie intake was consumed at dinner. Of note, in some of the European countries, like in France and Italy, where they have lower prevalence of obesity, diabetes, and other cardiometabolic diseases, majority of their daily calorie intake is consumed earlier in the day. But what we found in this study was that US adults consistently ate more in the evening over the past 15 years. So maybe this is something that when we design strategies to battle the cardiometabolic diseases, maybe by looking at timing of intake and within the distribution patterns of calorie and macronutrient, we can design some more effective strategies. I like this figure because it shows that even within population subgroups, we have heterogeneity in chrononutrition behaviors. So what you see here is the proportion of young adults shown in these blue bars, middle-aged red bars, and older adults that are categorized according to their eating duration into short, average, and long eating window categories. So, for example, as you can see, among adults, older adults, about 40% of them had an average eating duration of 11 to 12 hours, and others were split between those eating less than 10 hours a day and those eating more than 13 hours or more than 13 hours a day, basically having longer eating duration. And then as for the late night eaters, so as you can see here in this study, we observed that mainly young adults were late night eaters compared to the other age groups. So, well, what we did next, we determined the associations between various chrononutrition behaviors, but this time with muscle health. among older adults. And for this paper, we use data from our ongoing cohort study of muscle mobility and aging, or SOMA in short. Before moving forward, I'd like to acknowledge my PhD student, Ziling Mao, for doing an excellent job in data analysis for this paper. So as you can see here, in SOMA, we enrolled 879 participants at baseline. And here you see the inclusion criteria for a study. We included all the adults over 70, aged 70 and over, who had BMI between 18 to 40, who were able to complete a muscle biopsy and MR scan for us. Our study participants also were able to walk a quarter mile and did not have any active cancer or advanced chronic diseases at the time of recruitment. And for this study, we excluded 51 participants who did not have complete dietary data. So our final sample was 828 older adults, of whom 59% were women, 13% were Black individuals, and the mean age was 76 years. So for the dietary assessment in SOMA, we collected two automated self-administered 24-hour food recalls, or ASA-24, which was developed by the National Cancer Institute. And then during the dietary assessment, we collected detailed information on the types and amounts of all the food and beverages consumed, along with their exact clock time over the past 24 hours. And we also asked our study participants to self-identify their eating occasions for each item as breakfast, lunch, dinner, snack, or drinks. And as for the chrononutrition behaviors, we defined and extracted the same chrononutrition variables that I explained earlier for the enhanced study. So for the muscle assessment, as you can see, these are the muscle measures that we looked at in our study as our study outcomes, including muscle mass, which was determined by D3CR method, thigh muscle volume by MR scan, and then, as you can see, we had maximum grip strength and peak leg power as other outcomes. Now let's look at the results. So what you see here is the mean clock time of these chrononutrition behaviors. So on average, among our study participants, the mean eating window was 11 hours a day. And the time of first food intake, as you can see, was around 8:20 a.m. And the time of last intake was around 9:20 p.m. What we observed was that women participants had a shorter eating duration compared to men, and they had a later breakfast intake time compared to male participants. And as for the race, what we observed was that Black individuals had the shortest eating duration, and they also had the latest breakfast, as you can see it here, and the latest lunch time compared to the other race group. Well, what you see here is our participants' characteristics by the tertiles of eating duration. So under the first tertile, we had those with shorter eating window of about less than 10 hours a day. And then here we had participants with average eating duration of about 10 to 12 hours a day. And under the last tertile, the third one, we had those with the longer eating window of about more than 12 hours a day. And as you can see here, participants in the upper tertiles of eating window were predominantly men, white, and from Pittsburgh. And regardless of what was the eating window was, eating duration, so what our study participants were similar with regards to education, marital status, and health status. And as for body composition and physical function, well, as you can see, compared to those in the lower tertiles, participants with longer eating duration, they had significantly higher muscle mass. greater muscle volume. They also had greater grip strength and also leg power. They were also slightly more physically active compared to those with short eating duration, as you can see here. So first, we looked at the associations between these chrononutrition behaviors and muscle mass on your left and muscle volume on your right. So what we observed here was that eating window was positively associated with muscle mass. So basically here, every 1 hour increment in daily eating duration was associated with about 0.2 kilo greater muscle mass. We also observed a positive association between the time of last food or beverage consumption and muscle mass. So here what you see is that every hour later in the time of the last food intake was associated with about 0.3 kilogram greater muscle mass, and these associations were independent of age, sex, race, marital status, total calorie intake, physical activity level, weight, height, and the other covariates that you see here. We also, what I'm showing here is our model before adjusting for total protein intake, but in the paper, you can see the model after adjusting for protein intake, and even including protein as one of the covariates did not change our results, and eating window and last intake time were still significantly associated with muscle mass. As for the muscle volume, well, majority of our chrononutrition variables were associated with muscle volume in our unadjusted models. However, after adjusting for these covariates, we did not observe any significant association. Okay, so next we looked at the relationship between these chrononutrition behaviors and grip strength and leg power. So as you can see, among our studied chrononutrition variables, only the time of first intake was negatively associated with the grip strength after adjusting our models for these covariates. And then for the leg power, well, eating window, first and last intake time were associated with the leg power in our unadjusted models, but upon adjusting our models for these covariates, only eating window was marginally associated with the leg power in the fully adjusted model. Okay, so here the final conclusion was that longer eating window, later and earlier intake time were associated with greater muscle mass, a greater leg power, and higher grip strength in older adults. So what I like to emphasize here, there are 2 points that I like to emphasize in this slide. One is that our findings contradict the reported benefits of fasting diets, which are mainly supported by non-human studies or studies on young, physically active and healthy individuals that, as I mentioned, they're not representative of older adults population. And most of these fasting diets, fasting studies, they've looked at its effect on weight loss or fat mass loss. And existing evidence suggests that these fasting diets are not different from non-fasting diets in terms of promoting muscle gain. And the other point is that, as I mentioned, most of our studied chrononutrition behaviors were associated with muscle measures in our unadjusted models. However, upon further adjusting our models for various covariates, more specifically total calorie intake, those associations were not significant anymore. So considering that in this population, undernutrition rather than overnutrition is our primary concern because of its association with poor immune function, delayed wound healing, hospitalization, and mortality. So maybe encouraging these individuals to have adequate energy and nutrient intake, maybe at the right time, will help them to maintain their muscle health. However, of course, large-scale, multi-center clinical trials are needed in order to confirm the reported benefits of such dietary interventions. So beyond just the timing of food or beverage consumption, the nutrient content of what's ingested, like proteins, carbohydrates, fats at a specific times, which is referred to as nutrient timing, is also crucial, and it's important to consider. Again, in sport nutrition, we have some dietary recommendations with regards to the timing of nutrient intake, especially around the competition or during their competition, in order to enhance the performance of these of elite athletes, as I've included some examples here for you to see. However, there is no such recommendations for the nutrient timing for the general population and in our dietary recommendations. So what I'm showing here is the is one of the study that we did to determine the nutrient intake timing according to the self-reported eating occasions, snack, breakfast, lunch, dinner among US adults, again, using enhanced data. And what we observed here was that the highest intake of all macronutrients, including dietary proteins, fat, and carbohydrate, is skewed towards dinner. And this is a pattern consistent with the distribution of calorie intake, with the peak calorie intake that we previously observed at dinner. So there are many ways that we can look at nutrient timing. For example, we can use trajectory models, or we can define them according to the clock time of intake, or even here, we define them based on the self-reported eating occasions. Or we can actually determine their within the distribution patterns to see if their pattern of intake is even or whether it's skewed towards one of the meals. So what I'm showing in this slide and in the following slide is a study that we previously did by looking at the within the distribution patterns of protein intake. And why this is important, why it is important to know the distribution patterns of protein intake within 24 hours. Well, there are studies, small-scale studies, mainly animal studies, that they've shown that even distribution patterns of protein intake throughout the day, basically eating the same amount of protein at breakfast, same amount at lunch, and same at dinner, having this even pattern can simulate muscle protein synthesis in older adults more effectively compared to the skewed pattern that I'm showing here. And this is a pattern, actually, it's a common pattern that most of us follow. We have a carb-dense breakfast, and then we consume majority of our daily protein intake at dinner. So our group, for the first time, determined the link between protein distribution and lean mass in a large cohort of community living older adults. And here, our hypothesis was that even within the distribution patterns of protein intake, independent of how much you've consumed proteins, is associated with greater lean mass and reduced rate of decline in lean mass over time. Okay, so for this study, we use data from the Quebec longitudinal study on nutrition and aging, NUAGE in short. It's a Canadian cohort. For this study, we included all the participants who had body composition and dietary data assessed at baseline and after 2 years. So our final sample in this study was 712 older men and women. So as for the dietary assessment, in NUAGE, they collected 6 24-hour food recalls that help us to determine the amount and the distribution of protein intake, because we had the time as precisely as we could, and then the assessed body composition by DEXA to determine total lean mass and fat mass. And these are the covariates, actually, that we included in our statistical models. So this is how we determine protein distribution. We use the coefficient of variation of protein intake, or protein CV, which is this formula. This is basically a standard deviation of grams of protein consumed at breakfast, lunch, dinner, divided by total protein intake. And once you plug your numbers in this formula, you get a number. The closer the number to 0, it means that you have an even this pattern, a distribution pattern of protein intake throughout the day. And the higher the number, basically, it's just you're having an uneven pattern. So what I'm showing here is the baseline data for men, the blue bars, and women, the red bars, for the total daily protein intake on the right, and then protein intake grams per meal per day for the distribution. So as you can see, what we observed was that male participants had significantly higher protein intake compared to female. And as for the distribution, again, this is a Canadian cohort, we observed the same skewed pattern as in US adults, with a majority of protein intake being consumed at dinner and lunch, with breakfast only contributing to about 10% of daily protein intake. And it's worth to mention that our study participants kept eating the same amount of protein with the same pattern at baseline and throughout the follow-up period. So basically, this is showing same thing. This here we have the grams of protein intake ingested at breakfast, lunch, dinner. But this time I'm showing the data according to the quartiles of protein distribution, or protein CV. As you can see, participants at Q4, basically these white bars, they had the most skewed intake towards dinner, while those at Q1, this black bar, you can see that they almost consume the same amount of protein at lunch and dinner. And we observed the same pattern in men and women. So we can say that how much protein they consumed at breakfast and dinner mainly determine the distribution patterns of protein intake throughout the day. But what was interesting was that regardless of what was the distribution of protein, is basically at the end of the day, our study participant, it seems like they consume the same amount of protein, almost the same amount of protein across the quartiles of protein distribution. So what we did in this study, we first determined the cross-sectional associations between protein distribution and lean mass. So here on the x-axis, I have quartiles of protein distribution. So under the first quartiles, we have participants with even patterns, and then moving to Q4, you see participants with uneven pattern of protein intake. And then on the y-axis, it's the lean mass in kilo for the baseline data, the white bars, and the second-year follow-up data shown in gray bars. So as you can see, even distribution patterns of protein intake was associated with greater muscle mass in both men and women, both at baseline and after 2 years. And these associations were independent of the amount of protein intake, and also these covariates that you see here. Next, we determine the relationship between protein distribution and rate of decline in lean mass over 2 years. So among NUASH participants, we observed about 2.5% decline in lean mass after 2 years in men, and 2% lean mass loss in women. But as you can see, the amount of loss is sort of similar across quartiles of protein distribution. So here the conclusion was that protein distribution was cross-sectionally associated with greater lean mass, but it was not associated with a reduced rate of decline in lean mass over time. And then we looked at the relationship between protein distribution, but this time we used physical function as our outcome. And our hypothesis was that evenly distributed protein intake, again, independent of the quantity, is associated with better physical performance cross-sectionally and a reduced rate of decline over 3 years. in our mixed model. So again, for this paper, we use data from the NUAGE, but this time we included all study participants, 1,793, that we had their physical function assessed, and along with these variables at baseline after year 1, year 2, and year 3 during the follow-up period. So how we determine physical function here, we grouped physical function into 2 functional composite score. As you can see here, one was the muscle strength score, which was calculated as the sum of these 3 muscle strength tests, including hand grip strength, arm and leg strength. And then we created the mobility composite score, which was calculated as the sum of these 4 mobility function tests, including time up and go, chair stand, and gait speed. And these, again, were the covariates that we considered and included in our mixed model analysis. So just like lean mass, here, we first looked at the cross-sectional association between protein CV or protein distribution and muscle strength. So what you see here, again, I have on the x-axis quartiles of protein distribution, and then here on the y-axis is the muscle strength score. And I'm showing data for men, the black bars, and women, the white. So as you can see here, even distribution pattern of protein intake was associated with greater muscle strength in both men and women, and this association again was independent of the amount of protein intake. So what you're seeing here is the baseline data. We also observed the same cross-sectional associations for both men and women at year 1, year 2, and year 3. And again, these associations were independent of the quantity of protein intake. Then we looked at the longitudinal association between protein CV and 3-year changes in muscle strength. So as you can see here, we observed 20% decline in muscle strength in men and 18% in women, but the amount of loss in muscle strength is similar across quartiles of protein distribution. So again, the conclusion was that cross-sectionally, protein CV is associated with greater muscle strength, but it was not associated with a reduced rate of decline in muscle strength. And then as for the mobility function, unfortunately, protein CV was not associated both cross-sectionally and longitudinally with mobility function. So here I'm showing one of the mobility tests, chair stand. This is baseline data, and as you can see, it's just there's not any significant association between protein distribution and mobility function. So this is the last section of my today's presentation. I wanted to focus on the underlying mechanisms by which diet influence muscle health. Here I've included some potential mechanisms that various measures of diet, including quality, quantity, and time of intake, can influence muscle health. And these potential mechanisms are that I have here. One of them is the gut microbiome, and the other one is inflammation, which is the low-grade chronic inflammation in aging. And why this is important to know more about these mechanisms is basically knowing how how these mechanisms actually mediate the relationship between diet and muscle health will help us to better design dietary strategies to optimize muscle mass and muscle function. The thing that I'd like to say here is that in order to see how timing of intake influence these mediators, we first need to establish how the actual intake, which is the quality or quantity of food or beverage consumption or nutrient, can influence these mediators, and then determine how the chrononutrition can influence gut microbiome and inflammation. Basically, here we need to move from a bigger picture to a detailed one. So what we've done so far as our first step was to determine the association between quantity and quality of dietary protein intake and gut microbiome composition among community living older adults. And here our hypothesis was that a greater amount of protein intake would be associated with a greater gut microbiome diversity in older adults. So for this study, we use data from the osteoporotic fractures in men, Mr. Oz study on community living older men. So in Mr. Oz, they collected dietary data during visit 4 using the Omnigen gut stool collection kit, and then they determined gut microbiome composition by 16S RNA sequencing, looking at the V4 region. And in this study, we extracted total protein intake and its sources, whether it was from plant or animal sources, from the collected food frequency questionnaire. So the sample size for this, you can also see the exclusion criteria. We removed those without having dietary data and using antibiotics, probiotics, with very low or high energy intake, and those who self-reported that of having following a higher protein diet. So our final sample size for this paper was 775 participants. So we first determined differences in the gut microbiome richness and evenness according to the quartiles of protein intake. This is total protein. And as you see here, we observed that higher protein intake was associated with greater Shannon index and Chow-1 index, which are measures of gut microbiome diversity. And then we compare differences in alpha diversity measures, but this time according to the sources of protein intake. So here we observe in the upper panel, you see the vegetable protein, and in the lower panel, I have the animal protein. So similar to the amount of protein intake, again, higher intake of protein from either vegetable or animal sources was associated with greater gut microbiome diversity. And in our last step, we observed that, there are differences in gut microbiome diversity according to protein intake. So let's see what are the taxa that are responsible for the differences that we observed in gut microbiome diversity. So here we did differential abundance analyses using ANCOM BCR package. What you see is the pairwise differential abundance analysis at genus level. Here we compared the second quartile of total protein intake versus the first quartile. This is the third quartile versus the first, and these are those with the highest amount of protein intake at Q4 versus those with the lowest protein intake at Q1. So what we observed here was that the taxa that I put red circles around them were significantly more abundant in people with higher protein intake compared to those with lower protein intake. So now that we've established the link between quantity and quality of protein intake and gut microbiome composition, our next step is to see how within the distribution patterns of protein intake is associated with the gut microbiome diversity. But for this future study, we're planning to use data from the stoma cohort that we have the time of intake. So hopefully in the near future, maybe in other seminars, I'll be able to share the results with you. So this is the final conclusion and the broad implications of the research that I presented here. So basically our findings highlight the role of chrononutrition and basically how meal timing is one of the key factors influencing changes in muscle mass and function with aging. So this can aid in the development of precision nutrition strategies for older adults by guiding them what and when to eat, in order to meet nutrient needs, prevent sarcopenia, and to promote health. So with that, I'd like to thank all the people who helped me in these projects, especially Dr. Ann Newman, who's one of the main PIs of the SOMA cohort, also investigators at McGill University, my alma mater, for all the support and help that I received from them during some of the projects that I presented, and also National Institute on Aging for supporting my research. And thank you all for your attention. I'm now ready to take any questions that you guys might have.
Thank you for a very interesting talk. I guess I'm going to start, take the prerogative to ask the first question. And you noted when you're looking at the protein distribution that after you accounted for total protein intake, it didn't affect any of the associations. But was there a substantial portion of these participants who had inadequate intakes? And if so, were you able to account for that just by looking?
Yeah, so the mean protein intake was 0.8, which is sort of related to the RDA for protein, which is the same. But you're right, actually, in some of the cohort data that in some of the studies that I used, for example, in Mester Oss, the median protein intake is even less than 0.8, is 0.7. Gram per kilogram of the body weight per day. But I think what's interesting about the distribution is that just showing that even, let's say, even these older people are not able to consume more protein, higher protein, what are the feasible ways that we can help them to still maintain their muscle mass and function? Maybe just observing the skewed pattern in protein intake, maybe just asking them to cut some of the protein that they consume at dinner and add that to their breakfast meal. Maybe it's one of the ways helping them to maintain their muscle mass. Of course, we need to encourage them to consume more proteins. There are studies showing that even 0.8, it's not enough for older people because of the metabolic resistance. It's not enough to stimulate muscle protein synthesis in these people. So I think as a dietitian, trying to figure out different ways that we can help these people, because for some of older adults, I think it's just difficult to ask them to eat more or to increase their protein intake, like they have dental issues. It's just hard for them to cook or to buy these meals, again, based on individual cases, see if they need protein supplements, and if we can play with the timing of intake, helping them to maintain their muscle health. We have to be creative. It's just different ways to in helping them.
Thank you. Okay, so the next question comes from Larissa Constantino, and she asked, does your results suggest that TRE is not recommended for older adults?
Yes, but as I mentioned, we need more studies, because what I presented was a cross-sectional analysis. We are actually doing the longitudinal analysis now, because it's not to better establish the cause and effect relationship. We need to basically see if we see this relationship in other studies and in long term. But what we observed here was that, again, going with the movement of precision nutrition and trying to tailor dietary recommendations according to the individual needs, I think in older people, encouraging them to have short eating window without having enough evidence, and most of our evidence are from animal studies or studies on young, as I mentioned, physically active individuals that are not representative. I think we need more evidence before encouraging older people to follow calorie-restricted diet for the longevity, that it's a thing now, and also follow the fasting diets.
The next question comes from Saidas, who
Hi, Sai.
She says, nice talk. Did you look at calorie and nutrient adequacy in those with a shorter eating interval and lower muscle mass versus those with a longer eating interval and higher muscle mass?
We did not. So maybe, yeah, that's another thing that we can look at. But, just because of losing power when we categorize participants into these categories. So we try to control for, let's say, total calorie intake or, like the important factors or variables influencing the results. But yeah, that's a great suggestion. It's just I'm all for showing the heterogeneity in a population, and I know that even within older adults population, as we showed in NHANES data, we have some people with long eating duration with average, and we also have those with short eating duration, and that'd be great to see if there is a link between the differences that we observe in eating duration with the muscle outcomes. Yeah, sure.
Next question is from Jiandan Wang, who asks, you have showed that the eating window was associated with muscle mass in the older adults. Since a number of old adults take many dietary supplements, for example, he lists some Coke, Q10, omega-3, vitamin D, creatine, etc. I was wondering if timing to take the supplements may play a role. Example, decreased or increased or no effect on muscle mass and health.
Yeah, so basically, like I'm an epidemiologist, so the type of studies that we do is just trying our best to control for all the, like the factors influencing our study outcomes. So in this study, we, as I mentioned, we collected dietary data using ASA24. In ASA24, participants are given the option to report if they're taking any supplements or not. And we did not actually control for that, but we did control analysis for diet quality and for total calorie intake. So yes, of course, like what I presented is just the associations, is not the causal relationship, but there are many factors, even like the ones that we may have not considered influencing the association. So that's why I think that we need longer term follow-ups to do we need clinical trials, cohort studies to better establish the link between eating window and muscle health in older people. But like we try our best to control for the variables that we thought they're influencing the muscle. They're actually correlated with the muscle, with the outcome variable and dependent variable. Yeah, but yeah. Maybe like a well-controlled clinical trial will help us to better address if the eating duration indeed is influencing muscle health in older people.
The next question is from Sarah Booth, who
Hi, Sarah.
Compliments you on your talk. Wonderful talk. How do you account for beverages in your eating window and frequency measures?
How we determine beverages?
How do you account for beverages?
We actually we looked at the total calorie intake if it was more than 0 calorie, either from any food or beverages, we considered that item as the first or last intake time. So even like any beverages having some calorie content more than 0, we considered that intake as the first and last.
Next question comes from
Alex. Can I just add one more thing? So
yes.
with these chronic nutrition behaviors, unfortunately, we don't have a definite, or at least like a definition accepted by, investigators or like people in the chronic nutrition field. I know Dr. Santonge at Columbia published a paper in Circulation trying to define these chrononutrition variables so that people that are studying the link between chrononutrition variables and outcomes, we're using the same definition for that. Unfortunately, it's a new field, and for most of these chrononutrition behaviors, we don't have a agreed upon definition for, or even I've seen that in some of the studies, they're defining the intake as the contribution of any food or beverages more than 2 calorie. to the overall daily eating. So it's just what we used in this study was if the content of any food or beverages, if the calorie content of any food or beverages was more than 0 calorie, we considered that as an intake for to define the first and last meal.
Okay. Alice Lichtenstein asks, with regard to the effect of protein intake on gut microbiota diversity, besides different absolute protein intakes, could some of the diversity be attributed to the alternate micronutrients, for example, fat or carbohydrate quality?
Yeah. So I didn't show actually beta diversity measures. I was just worried about the time, but we also observed differences in beta diversity measures based on protein intake. And in those analyses, we try our best to control for potential covariates, including total calorie intake, or, like I can't remember, but I think we also controlled for maybe like diet quality as a surrogate measure. But I think controlling for total calorie intake at least can help us, is a surrogate measure of the intake of other nutrients who might influence gut microbiome composition. We also actually control for fiber intake as well, because knowing that based on the literature, it significantly influenced the gut microbiome composition. Unfortunately, I can't remember the details of all the covariates that we include in our models, but I think total calorie intake and fiber were definitely one of the covariates that we included in our diversity models.
Okay, next question is from Roger Fielding.
Hi, Roger.
He says, again, really nice presentation, he says.
Thanks.
How do you think about data from the SOMA study where you observe significant relationships with D3 creatine and protein intake variability, but not with muscle volume? That's the first part of the question. The second part, go with that now. I guess I'll ask the second part later.
Yeah, so I think I discussed that in the discussion section of the paper. So saying that because the D3CR method for the assessment of muscle was for the total muscle mass, but the MR scan is just a thigh muscle. So I know MR is also the gold standard for the assessment of muscle tissues, but maybe because it's just a part of the body, it's not representative of the total muscle mass in the body. So maybe that's one of the reasons that we observed the significant association between core nutrition behaviors and muscle mass assessed by D3CR, but not with the MR scan. Maybe next step is to see if there is a link between core nutrition and muscle volume, like total muscle volume, assessing that from the MR scans. Because I think we have the MR scan from like head to toe of these participants.
Okay, and the second part of his question, which will probably be the last given the time, he asks also, where do the mean kcal and protein intakes lie in the SOMA study cohort?
I think I can't remember, but I think it was around 0.8. I think it was around 0.8 gram per kilogram of the body weight.
Okay. All right. Well, again, thank you very much for a wonderful presentation.
My pleasure, of course.
Have a good day.
Have a nice day. Bye-bye.
Bye.